This is the pre-lab talk for experiment number 3 for Chem 120 lab, freezing points. The goals of this experiment are to observe and explore phase changes, namely the melting and freezing point equilibrium. We'll also explore how the freezing point of a liquid is affected by different solutes. To do this, we will use the standard deviation of the mean to determine if values, namely the freezing points of different solutions, are significantly different. Finally, this lab makes use of thinking and understanding skill number 3. Observations lead to explanations, conclusions, and/or questions. For today's safety pause, we note that dry ice is particularly hazardous. The two main hazards associated with dry ice are that it is extremely cold, which can cause cryogenic-like burns. Dry ice is also an asphyxiation hazard, as it produces carbon dioxide gas, which can make it difficult to breathe if in a pure CO2 environment. To mitigate these hazards, we will make sure that we wear gloves when handling dry ice. We'll also only handle the dry ice with either scupula, forceps, or tongs. And we'll also keep dry ice out of sinks where it can combine with water to form large water/CO2 clouds, which can cause an asphyxiation hazard. It can also cause damage to the plumbing in the sinks. For waste disposal in this experiment, all of the solutions can be flushed down the drain with plenty of running water, and any unused dry ice should just simply be returned to the cooler where it was provided. In this experiment, we're mainly going to be studying a phase change equilibrium. On the right is what we call a heating curve, where we plot the temperature of your substance versus the heat that's added to the substance. This heating curve is for water, which we'll be studying in this experiment. We note that at the freezing point of water, 0 degrees Celsius or 273 Kelvin, that both liquid water and solid water are at equilibrium with one another, meaning that freezing and melting occur at the same rate at the freezing point temperature of water. We also note that at the freezing point, the temperature remains constant, although heat is being added to the substance. This is because all of the heat goes into causing the phase change, where you have to break a lot of intermolecular forces, instead of going into changing the temperature of the substance. These two criteria, looking for the presence of both the solid and liquid phase and looking for no change in temperature, are both what we'll be looking for when determining when our solutions or our pure water are at their freezing points. The other important concept that we'll look at is the concept of freezing point depression. You might have talked about in lecture how when a non-volatile solid is added to or dissolved in a solvent, the resulting solution has a lower freezing point than the freezing point of the pure solvent. This is one of the 4 colligative properties of solutions that only depend on the amount of substance added and not the identity of the substance added. We note that the change in the freezing point temperature, delta Tf, depends on 3 factors. The first factor, I, is called the Van 't Hoff factor, which tells us the number of moles of solute of particles that are formed per mole of solute added. For sodium chloride and ammonium chloride, which are used in this experiment, I is equal to 2. That means that 2 moles of particles are formed when 1 mole of each salt dissolves in water. Kf is known as the freezing point depression constant and is in units of degrees Celsius per molal. The freezing point depression constant solely depends on the solvent that is part of the solution. Since we're working with water, that constant should be the same for any solution formed in this experiment. The last variable is the molality of the solution. The molality is one form of concentration and is equal to moles of solute dissolved per kilogram of solvent. We're also going to attempt to keep the molality as constant as possible. However, this might not be the case, as you'll see later in the experiment. Here are a few procedural notes for this experiment. More will be discussed in the lab video. In this experiment, we'll be using the temperature sensor and LagerPro software. You can look at Appendix C to see how the temperature sensor and LagerPro software work. But for now, remember that the temperature sensor must be plugged into a LabQuest Mini, which serves as an interface between the computer and the sensor itself. In LagerPro, we'll be using what's known as time-based measurement, where a temperature will be measured for every increment of time as indicated in our data collection mode setup. This will allow us to look at the temperature as we add dry ice to our solution in our solvent or solution in part 1 and adding salt to our ice in part 2. We'll be able to see where the temperature remains constant, one of the criteria for determining if our solution or our sample is at the freezing point. In this experiment, we'll need to take our dry ice, which sometimes comes in larger chunks, and crush it up into almost a powder to avoid explosions that happen when water freezes around a large chunk of dry ice, creating a closed system, which can result in a pressure explosion. Therefore, we will crush the dry ice into a powder-like consistency. We'll perform our experiment in a calorimeter, which, as you might remember from Gen Chem 1, consists of a pair of styrofoam cups nested inside one another. The only difference between this experiment and the other experiments you did in Gen Chem 1 is that we won't be using a lid so that we can observe what is happening inside the cup. You might also remember that we need to stabilize our calorimeter by placing it in a 400 milliliter beaker. It is also important that as we collect data that we create a baseline before we start adding our dry ice. That way we can look at the difference in temperature between starting the experiment and ending our experiment. The last thing we want to talk about is looking at class data and the use of the standard deviation of the mean. For this experiment, although we're doing this experiment online, you will be able to look at and analyze some class data for the freezing points of sodium chloride and ammonium chloride, and for the percent of ice that melts in part 2. As a reminder, We typically average class data to get a more accurate picture of the phenomenon that we want to study. In the picture on the right is a histogram of density data collected from experiment 2 from GenChem 1 lab. The red X represents the average in the data. In order to analyze our data, we want to take into account both the average value and the spread in the data. We can use the statistical spread in the data, also known as the standard deviation, to help us do this. In this picture, the standard deviation is represented by the range that defined by the blue arrow. You can see that most of the data here exists in that range. You can calculate the standard deviation by using your calculator or using Microsoft Excel. However, we want to know what the uncertainty is in our average value. We can do this by taking the standard deviation and dividing it by the square root of the number of trials. We call this value the standard deviation of the mean, which gives us an uncertainty in our average value. Therefore, when you calculate your standard deviation using either Excel or your calculator, you simply need to take that number and divide it by the square root of the number of trials. This tells us that as we increase the number of trials, the more certain our average becomes, which is the main reason why we want to pool class data, so that we're more certain of our measured value. When you report your averages, you want to report them using the standard deviation of the mean in the following manner. Take your standard deviation of the mean and only quote it to one significant figure. We do this because typically in our measurements, there's one uncertain digit. Then, when you quote your average, make sure that the average is rounded to the same number of significant figures or same number of decimal places as your standard deviation of the mean. When comparing two values, along with the standard deviation of the mean, you can tell that they're significantly different if those values, along with the range created by the standard deviation of the mean, do not overlap. If they don't overlap, you can say that those numbers are significantly different. However, if they do overlap, you cannot say with certainty whether the averages are significantly different.